virology, the most amazing subject matter ever. Not that I'm biased at all, in the very least. If you want to know more about some of these things, these are kind of the ways that you can contact me. We will be actually covering all of the things that are on this slide, assuming I can get my pointer to work. These guys, which are a really fascinating plant virus, cowpea chlorotic mosaic virus, a virus that very, very near and dear to my heart because I actually discovered this particular virus, the STIV virus, the one that a couple of you know way more about than you probably ever want to admit. SSV1, this is what we mostly work with in my lab. Adenovirus, and then maybe bacteriophage T4 if we get a chance to talk about that later. So where do you find me? Hanging out with my viruses. This is, some people call me the virus head or the virus capsid. 466 Science Research and Teaching Center. Was it STRC, SRTC? I can never remember which one is right. My office hours are right after class. If there's nobody in here right after our class, which I hope, I didn't actually check, we'll just have office hours here. It's way easier than tromping across campus. If, however, there's another class in here, we'll stay here until they kick us out, and then sort of move slowly in that direction. If for some reason you can't, Ian brought salad for us. Thank you, Ian.
You get your own.
So if for some reason you're looking for me there and can't find me during my office hours, here is probably where I'm going to be. You can reach me by email. I'm ridiculously email dependent. Again, a number of you know this. kstedman@pdx.edu. You can try sending emails to the D2L email. I will usually forget to check if there's anything there. If you really want to talk to my voicemail, call my voicemail. But very, very rarely I'm actually going to pick up the phone. I do post on Facebook, and actually it's kind of a funny story about Facebook. My lab actually created this Facebook page for me when I was on sabbatical because they said, you need a Facebook page. So I did a Facebook page. I also have all my lectures on YouTube. You can go and check those out anytime you like, if you really want to. My website is ridiculously out of date, and about 3 months ago I got dragged kicking and screaming onto Twitter. And now I can't imagine not having Twitter. It's actually kind of frightening how quickly that happens. Plus, I got, I think, one of the coolest ever handles for this, Extreme Virus Prof. So very easy for you to find me as far as these different places, or just look for the big white virus, as the case may be. So a couple of things people have asked me about as far as this class is concerned. How many of you have not had molecular biology? And I don't know your names, so fine. We can chat about that. That's good. I will assume that you know things that I talked about in molecular biology last term, or presumably Dr. Singer has talked about in molecular biology. A couple of years ago, a couple of students started writing down every time I said, remember from molecular biology. I think they got up to about 60 in the term. So a lot of stuff as far as that's concerned. Cell biology. How many of you have not taken cell biology? Okay, that's not a huge problem. We can definitely get past that. The main things we're going to talk about in terms of cell biology have to do with signaling, but in a very basic sense, and just subcellular organelles. So a lot of the stuff that hopefully you got in principles should be sufficient cell biology-wise. If any of you have questions about that, please come and talk to me. Probably after class is the right time to do that. But I've had plenty of people who haven't taken cell biology or are taking cell biology simultaneously with this course, and they do just fine. Also had people who've done that and not done so well, but so be it. So what are we going to talk about? Viruses. I could talk about viruses, and actually most of you who know me know that I can talk about viruses for a really, really, really long time. Unfortunately, we only have 10 weeks and 3 lectures a week to do that. So I'm really going to concentrate on molecular virology. I'm not going to talk about virus pathogenesis very much. There are some interesting exceptions to that, and mostly has to do with vaccines, so you don't have to worry about any of this viral pathogenesis stuff. So it's really going to be molecular virology at a pretty basic overview kind of level. Again, we could spend literally years talking about viruses, and I have. But the main things we're going to talk about here really have to do with sort of, again, these main classes of viruses. People will argue whether there are 6 or 7, but that's just at the very molecular basis. We've got DNA viruses, we've got RNA viruses, and we've got retroviruses. And then we've got these bizarre chimeric viruses that hopefully I'll have a chance to talk about later in the term. But those are the 3 sort of big ones. Start out with bacteriophage, some of our favorite. Anybody have their giant microbe bacteriophage? Bacteriophage T4. This is one of the best giant microbes ever. I'm trying to get them to make one of these too, but they haven't done it yet. And then we will talk about HIV, which is one of the more complex viruses. We'll also talk about some of the giant viruses, the mimiviruses, which have genomes that are larger than a number of microbial genomes. Actually, even one virus that has a genome larger than some of the smallest eukaryotic genomes, which is really pretty mind-blowing, at least it is to me. But the very majority of things we'll talk about is virus replication, and virus replication in a very general sense. So how do they make more of their genome? How do they make more of their capsid? And a little tiny bit of what they do to the cells that they're infecting. But mostly that's because they need to make more of themselves. So it's really going to be a lot about virus replication. So that is your genome replication. Genome, again, can be DNA or RNA, which is one of the really fascinating things about viruses. How do you make the proteins that need to come together to A, make more of your genome, and B, make more of the capsid, the part around the outside, what I also like to call the bag around the nucleic acids, and then putting everything together. And we're going to talk about this basically for every single set of viruses that we talk about, as far as each of these various different things. How is it making more of its genome? How is it making its capsid in terms of making the proteins, and also putting all of those things together? So we know the least about this last one here, actually, the assembly process, and that's one of the things that my lab is particularly interested in. Unfortunately, we have to do this evaluation thing. So those of you who are in molecular biology, check, first time you said it, there will be 2 midterms and 1 final, just like we had in molecular biology last term. Not, of course, the same days. Midterms will be here. And there was a mistake that in the first syllabus that I posted, I actually think I put down Monday rather than Wednesday. For the second midterm, the 17th of May, Monday the 24th of April. If you know there are going to be problems at those times, let me know ASAP, i.e. by the end of this week. If you have DRC that we'll talk about a little bit later, make sure that you schedule those times as quickly as possible. Final will be Tuesday at 8 o'clock in the morning, way too early for most of us. Also in here, that's the regular exam time. So it's exam week, Tuesday morning, so not Monday, Wednesday, or Friday. The exams will probably be just 50 multiple choice questions. I've been toying with the idea of having a couple of fill in the blank, short answer kinds of things. You think that's a good idea? Yes? No? No? Yes? We'll see. Maybe I'll try a couple, but not very many, because I've got to grade them all on the first midterm, but not, and we'll see how it goes further on. Yeah?
If you did that, would we be able to use the same big blue Scantron, just the back writing part, or would we be filling out other?
So Ian always asks the hardest questions, which is great. So sorry for the people who I know you. I will pick on you for the rest of the term, so consider yourselves warned. I'm not sure is the answer to that question, whether just use blue Scantron in the back or whether I'll just have like a tear-off page at the end of the exam, if I decide that I'm going to do that. I'm still, again, kind of, torn, and I didn't even mean that. So to see what we're going to do. Examples on D2L, I post all of my old exams there because I let you take your exams with you, and so that'll give you a really good idea what kinds of questions I am likely to ask as far as the rest of the term is concerned. Yeah? Okay, good. Next point, everyone's favorite, clickers. We will have clicker questions. I didn't set them up today, so we don't have any clicker questions today. How many of you do not have a clicker? Yes, so personally you will need a clicker for this course. They're about 20, 25 bucks. You can get them on Amazon. Used is fine. The old version is totally fine as well. But you will need those as of the second week. They will be 10% of your final score. And I will be spreading those clicker questions out throughout the lecture. I'm not going to do them all at the beginning and then have the people leave right afterwards. So they will be spread out through the whole class. I don't use reef polling for all kinds of different reasons that if you want to talk about, we can do after class. Make sure you register those on D2L. Not on D2L, sorry. This should be iClicker.com. And then, excuse me, iClicker.com to register them. That's, again, didn't update this from last year, obviously. Registering on D2L is really a pain, and it doesn't work very well, unfortunately. So iClicker is a better place to do that by the first midterm. If, for some reason, somebody's got a ton of clicker points and I don't have a name connected to that clicker, I will try and reach out to you and say, hey, is this you? Or not. I won't tell you how many clicker points you have at that point, though. How do I grade? Again, those of you who are in molecular biology last term, can ignore this. It's exactly the way that I've always done it. So I normalize the high score, whoever that person is, probably whoever she is, likely is not. That then ends up being where I scale everything from. And I'll let you know every midterm where that is. I only do final grading at the very, very end of the class, but I will set things up in such a way that that will be the final high score, and I will definitely let you know what that is. I usually round down a couple of points after that, just to see where the breakdowns are. A lot of people have asked, what about those annoying postbacks who come here and set the curve for everybody else? In molecular biology last term was a junior biology major who set the curve. Actually, it was above setting the curve, and I set the curve a little bit lower than that. So yes, there are postbacks, but they're not always the ones who are doing the best in our classes here. But of course, that's because the PSU students we've trained better than anyone else, at least in theory. As usual, exams are closed book, no time limit. Again, hopefully there's nobody in here after us and we'll stay here. If that is not the case, we'll just move outside and keep going. But because I do let people take their exams home with them, as soon as the first person turns in the exam, they've taken it and left the room. So nobody can start after that. So just make sure you're here on time. It's not such a big deal as far as the normal midterm times, as far as the final starting at 8, make sure that you're here. Again, DRC, make sure that you contact me ASAP. I haven't heard from anybody yet as far as that's concerned. Yeah, this is stuff I just have to put up here. I don't absolutely require attendance, although of course with clicker points means you probably will be here. But just in the past, people say they're always here. They usually get A's and B's. People who say that they're rarely here, sometimes get B's, but there are a lot of other scores that end up in there as well. And this is actually before I had a bunch of clicker questions. Makeup exams, you can basically read this. It's all posted. I actually posted this lecture on D2L. How many people pulled up the lecture already? Found it? Okay. Most people are molecular last term, probably. So you'll need lots of really good excuses for this. My standard excuse was actually last year, my dad died on Saturday, and I gave an exam on Monday. So this gives you a bit of an idea. Regrades, if you need them, make sure they come in within a week. I scan all of your Scantrons, so I've got all of that data if we happen to need it. And again, you can read all the rest of this stuff. How do you do well in this class? Main one is down here at the bottom. Ask lots of questions. That's by far and away the best way to do it. Textbook, I didn't bring this with us. You'll also notice that it's a fundamentals of molecular virology here. A couple of the reasons that I like the text. A, it's not ridiculously expensive. Most of the other ones are like $200 plus. The second one is that I really like the chapter overviews, which also go into key terms and glossaries. It also has some nice references if you are interested in looking at some of the background literature. Each of the chapters has a nice list of references at the end, so you can go and find most of those things that are in there. There are a few things that we will cover which are not directly in the textbook. I'll make sure that we've got extra readings that I'll post on D2L as far as that's concerned as well. Lecture notes, I will post all the PowerPoints, try and get them up by 10 p.m. the night before. If not, all the previous lectures are there, which I've based this year's lectures on as well, if you want to get those a little bit earlier. If for some reason I haven't posted them, this happens, again, as some of you know, send me an email first thing in the morning. And you go, Dr. Stedman, where are the lecture notes? So I will try and do that. All of the recordings of the lecture, notice I've got this wireless mic, even though I'm sure you could hear me in the back without. That's because I'm recording this, and hopefully, assuming that all the recording systems work, which they don't always, I will be posting my audio and then the video, which you see on the screen, on YouTube right after lecture, relatively right after lecture. So that's there. Also, all previous years' recordings are on my YouTube channel as well, so you can go and check those out. That's where it is. And again, ask lots of questions. So that's the sort of standard set of resources. Yeah?
I do have a question.
Sure.
Do you try to only base your exams on content discussed in this lecture, so like this class? Say that if you don't have time to get to a subject you did last year, would you not ask us about it? Or?
So basically, what is the material I'm going to have on exams? Would be another way just to paraphrase your question. When I make up, when I make up the exams, I just go back and look at my lecture notes, and the lecture notes that we get through in that particular class session. So if there's something which I think is particularly important that we didn't get to in one class section, I'll pick it up and we'll start it the next time. So if there's stuff that gets toward the end, which I often will sort of, that's that boring pathogenesis is, who cares, why they make you sick? Then what I'll do is just then leave that. That's not going to be something which is getting covered. And I have a nasty tendency to try and stuff too much in. Great way to slow me down. Ask questions. Exactly. So D2L, sort of mentioned already, syllabus is there, basically the stuff that I've already talked about. Also gives you a list of which subjects we're going to be covering at any particular time. One of the things I forgot to mention is with this particular textbook, it covers, first couple of chapters are general, but then it's sort of one chapter per family of viruses. That's just one way of doing a virology course. The other way is talking about DNA replication, RNA replication, etc. So the syllabus will give you kind of an idea where we stand with each of those. Lecture presentations, again, I'll try and post all the PowerPoints beforehand so that you can bring them to lecture and take notes on them. A lot of people who I've talked to in the past, those who've done very well in my class, have done that, brought them, taken notes on those afterwards. Old examinations and keys are also posted on D2L. Also a great way to study for exams is to go and look at those old exams. Supplemental materials, again, there'll be a few things that we cover here, just like molecular biology. That's the third time class today. He will talk about stuff that's moving in a really rapidly moving field. In fact, some of the stuff today that we're going to talk about, if we get there, which we probably will, has sort of changed what was in the textbook before. And so what is in lecture is usually right, although occasionally I'll go back and pick up my mail or open my inbox and find, oh, what I just said was wrong. And I will try and get you up to date on all of those things. Great way to ask about some of these issues is send me an email. I'll post it on D2L in terms of what was potentially in the past incorrect or was correct in the past but is now incorrect. I will try and make that clear. So that's where I put everything in the discussion forum. Any question that I get in my normal email, I'll just copy it, anonymize it, and post it to D2L. So usually if there's one person that has a question, it's not the only person. You're not the only person who has that question. So I will try and take care of that. And again, I occasionally will read the mail there, but I usually forget. So just send it to my normal PDX email, the kstedman@pdx.edu. I didn't bring my whole reference library with me. I would have probably needed a pickup truck to get it over here. A couple of things that are sort of really great references if for some reason you get excited about virology. I'm going to try. And want to learn more. Lots of different places that you can go and look. A previous textbook that I used to use that actually does this pattern of looking at the replication, DNA replication, RNA replication, transcription, etc., is the Flint et al. Principles of Virology textbook. There are actually 4 editions of this right now. That's the one that's $200 plus to actually get. It's a great reference for animal viruses, but unfortunately they don't cover my favorite kinds of viruses. So another reason that I'm not so wild about that particular textbook. There's a 2nd edition on reserve in the library, so you can go and check that out and take a look at some of the things that are in there. I actually do have a few of the figures in my slides, my PowerPoint slides, which I've taken from that textbook. Fields Virology. This is sort of the holy book of virology. I think it's 3 or 4 volumes now, each of them about 1,000 pages. One of the reasons I didn't bring it over. So also on Google Books has an older version of Fields Virology. Basically takes each of these individual viruses and breaks it down into a lot more detail if you're interested, and also covers a lot of the pathogenesis aspects of things. I mentioned that it's a fast-moving field. Journal Virology, Virology Science, and Nature often are publishing things that are virus-related, certainly Journal Virology and Virology, and even some of these, I don't know what people call them, vanity journals, I guess. Science and Nature also publish quite a lot of stuff. I have a pretty large library over in my office. If you're interested in borrowing things, I can certainly let you do that. The PSU library has a decent collection. If you're looking for something in the PSU library and it's been checked out, it's probably in my office. And then, find me in my office hours. There's a ton of stuff online as well. One of my favorite, competitors, colleagues, Vincent Racaniello, who has a blog, also a podcast that he does every week, TWIV, This Week in Virology. It's absolutely awesome. I, in fact, learn quite a bit when I listen to it every week, or try to every week. I'm a little behind. But he has a great virology blog. Also, he was the inspiration for me recording my lectures and posting them on YouTube. All of his lectures are recorded and posted on YouTube. He also has a Coursera course, one of these, massive online something course, MOOC. Yeah. But it's quite a good course as well. In fact, I've stolen, with his permission, a number of things from his lecture as well. As I mentioned before, or someone said TWIV, This Week in Virology. It's not just Vincent Racaniello, Kathy Spindler, who's at the University of Michigan, Alan Dove, who's a science journalist, ex-PhD student with Vincent Racaniello. Pardon? Elia Schechter actually does This Week in Microbiology, not This Week in Virology. And then the pox virus guy, who's named Rich Condit, who actually has a place in Sun River, and occasionally I see him at Mount Bachelor, but based originally out of Florida, now living in Texas. But it's a great, really fun. It's a little long, goes about an hour and a half to 2 hours. But if you've got a long commute in the max, something like that, great way to learn about all kinds of different viruses. They usually talk about 1 or 2 different current papers. I was one of the interviewees at one point a few years ago. In fact, based on that, I have 2 publications that came out last year because I was on this podcast. So great, wonderful stuff that they do there. Occasionally they get stuff wrong. I'll write them letters, tell them what they got wrong. Viper DB, when we talk about virus structures, this is a really nice server that tells you how to make virus structures, not like this one because we're not quite sure how this one fits together, but icosahedral structures like the head of this virus and the STIV viruses. I kind of tried to make a movie a few years ago. We'll probably take a look at the trailer here. I will warn you that it's not quite up to date. Viral zone, Has a lot of information on individual virus families, also a nice place to go, particularly given the structure of this course, individual virus families that we talk about in any given lecture. It's a great place to go and get sort of a orthogonal way of looking at any of the material that we particularly covered. And then just down here at the bottom, so I get a little bit out of date, the birthday party, 100 years of bacteriophage, or the first publications on bacteriophage, bacterial viruses, all available, all the talks here this was in San Diego now almost exactly 2 years ago, was in January if I remember correctly, including a talk of mine about sort of archaeoviruses, etc. What's some of the stuff that's not in the textbook or slightly different this term? We'll see if we have a chance to talk about bacteriophage T4, one of my favorite viruses. Why it didn't end up in our textbook, I'm not quite sure. I suspect someone was asked, and I probably know who that someone was, and they didn't get around to submitting a chapter. So we'll see if we have time to talk about that. This point, probably not. A couple of visiting lecturers, Alec Hirsch from OHSU, who's a flavivirus expert. What's everyone's flavy of the month? Flaviviruses? Any heard about Zika? Yes. So Zika is a flavivirus. And in fact, they work on Zika virus there as well. Although it's one of the least well-studied of the flaviviruses for historical reasons. Ryan Estep, who is actually a PSU grad, is working on herpes viruses, also at the VGTI. VGTI, by the way, is Vaccine and Gene Therapy Institute out at the West Campus of OHSU. And if I can convince George, who's a graduate student in my lab, to talk about the virus that he's working on, and someone else in this room who I won't point out immediately, but may force her to step up a little bit later on. Some of the other viruses from the Stedman lab, RDHV is that interesting virus that George would probably talk about. SSV, which is the one that's represented by this model here, STIV, and then also some updates on Ebola. Anybody remember Ebola? That was like 2 years ago, right? And Zika, which is a little bit more up to date. Yeah, Ian.
For the viruses from your lab, will we get like a paper or something to read ahead of time? Or?
Yeah, so the question is basically for these extra, the goodies, as it were, will I post some extra material? Yes, I will definitely do so. And most of that's actually already on D2L. So RDHV is already there. STIV, I think, is already there. Certainly, the latest SSV paper will be out really, really soon. Those of you who are in the molecular biology exam, one more of those, may remember that I got really excited because this guy's going to be on the cover of virology in about a month. So I will post that paper as well. So any more questions sort of about the general way the class is going to be run before I jump into how amazing viruses are? Okay, so let's start out with what we're going to cover for the next half hour, 35 minutes or so here. Start out with just a couple of virus definitions. This used to be really easy. Turns out it's actually gotten a lot harder in the last few years, what you call a virus, partly because of these giant viruses, partly because some of the weird viruses that we've found as well. Why should you study viruses? Well, because it's like the coolest things ever, but a couple of other reasons as well. One is that viruses are really everywhere, and one of my favorite lines is that viruses kind of have a bad rap. They're actually in our bloodstream, in our genomes, in the soil, in the air, etc. Most of them doing us no harm whatsoever. So most people study them because they make people sick. I'm interested in them for this whole diversity aspect of things and ubiquity. They're really absolutely everywhere. Then we'll talk a little bit about the history, of virology, particularly having to do with one of my favorite other things, vaccines. Vaccinate, vaccinate, vaccinate. Hopefully I will say vaccinate more than I say what you heard about in molecular biology last term. And anyone would like to talk more about vaccines, I'm more than happy to do that. How many of you had your flu shot this year? Those of you who haven't, think about how close you are to the next person right next to you, and if there are two people who haven't been vaccinated. Flu transmits about a meter really, really easily, and there's still quite a lot of flu circulating, particularly in Oregon. So we'll take a look at that. A little bit about pathogens. Yes, that's why people cared about viruses in the first place. And a lot of this virus diversity and ubiquity has only really been found out in the last probably 20 or 30 years. And then a little bit about how viruses were discovered. We may or may not get to virus detection today, and probably not to virus replication. So we'll talk a lot more about this figure here in just a second. So what is he didn't put virus on there, virion. So one of the very important aspects about viruses is the virion. This is what most people think about when they think about viruses. Well, this is a virus. No, this is a virion. This is the extracellular part of a virus. All viruses have to get inside a cell in order to be able to replicate. So when they're inside a cell, they don't have one of these things. So if you think about a whole virus life cycle, we can argue about whether viruses are alive later, then that's just the exterior part. And I will try and be careful about talking about virions versus viruses. I'm not always very good about this. But the virion is the extracellular part. And this is one of the parts which is really unique about the virus life cycle. It's got this extracellular thing, part of that particular life cycle. And this is what we often will talk about here. I like to think of these as bags of nucleic acid because they've got a genome. They've got something which is encapsulating that genome, or the bag around the outside. Very specialized genomes, very specialized bags. So here is just an example of two of the more common types. Got a nucleic acid genome on the inside, could be floating around, always has some proteins that are associated with it, either sort of on the outside with the nucleic acid on the inside, or directly associated with the nucleic acid here. So whenever we talk about the capsid, that's going to be the bag, the thing that's going around the outside. Some of these virions have lipids around them, which are called envelopes. Some of them don't. So we have naked viruses, those are lacking envelopes, or enveloped viruses. These envelopes almost always have a virus-specific protein that's sticking in those envelopes, but the lipids come from the cell. This is not a virus-specific, although we'll talk about some exceptions to that a little bit later on. Yeah?
Is that supposed to be the bacterial page T4 on the left, or kind of a representation of it?
Okay, so the question, is this supposed to be a particular virus? I'm sorry, I'm paraphrasing your question, but is this supposed to be a particular virus here? No. These are very generalized cases here. And these are two kinds of virions, but there are clearly other kinds of virions as well. So this is just a very general case. A lot of people have used with a lipid coating, without a lipid coating as being one way to sort of start classifying some of these viruses. And T4 actually does not have a lipid coating that's associated with it. So this is to kind of beat home this point. Ceci n'est pas un virus. Anyone get the reference? I put a little reference up here at the top. So this is not a pipe. Anyone know the artists who did this? Magritte, René Magritte. But anyone know who this person is? Patrick Forterre, one of my colleagues working on archaeal viruses. But he's the one who came up with this ceci n'est pas un virus. And basically the whole idea here is, just as I mentioned before, this is the virion, it's not the virus. So just something to bear in mind. And again, this is one of my favorite virions here as well. So that's your virion, but what about viruses? How are you going to define a virus? The standard definition, and again, anyone who's had that other course that I have taught in the past knows anytime it's in red and in quotes, you need to be very careful about what that is. But the original definition of viruses was a very small, infectious, obligate intracellular parasite. Well, very small, we know now, because of the giant viruses, is definitely not true. But the infectious, obligate intracellular, yeah, that's not too bad. Infectious, it's got to get inside another cell in order to be able to replicate. Parasite, I probably could have put in red here as well, because there are a number of viruses that are clearly good for their hosts. And again, we'll talk about a few of those as we move on through the rest of the course. However, all of these viruses do need some kind of basically metabolism and precursors. The virus in and of itself is not independently able to metabolize. One of the things that we will see, however, is that they often change cellular metabolism quite a lot. So precursors, usually the precursors here are the standard monomers that, again, we've talked about in the past, your nucleotides, your amino acids. They need energy. Probably one of the things that could be really emphasized here is the need for translation. So far, no viruses have been found that have their own translation machinery, at least not a complete amount of the translation machinery. And then, turns out they all need membranes as well, but that's probably because of this energy component. And pretty much always, in order to make energy, you need some way of separating things. Virus particles, I should say virions here, are not reused. And that's one of the things which really differentiates viruses quite well from cellular life. Cells are going to divide. They're going to take part of what they started with when they divide each time. Viruses, they lose their virion completely. That virion is never reused. So a very different way of undergoing replication, and that means that you don't have one virus that splits apart in the middle and becomes two. So a very different replication mechanism. Have a DNA or an RNA genome. Again, he's put it in red. What does he mean? That's because there are a few virions which actually encapsulate some RNA and some DNA in them. Mostly it's usually little short pieces of RNA and the rest of them being DNA. But there are a couple of examples of where you actually have both kinds of nucleic acid inside your bag in terms of the, yeah, sure.
So you're making the distinction, trying to make the distinction very clear between a virion and a virus.
Mm-hmm.
Is that because you'll have multiple different types of viruses with the same virion?
Okay, so the question here is, why am I trying to make this big differentiation here? It has more to do with the cycling. It's not because there's some genomes that all have exactly the same virion. There are some cases of that too, but we'll talk about those later. But basically here the idea is that the virion is only just one part of the replication cycle. And so because it's lost its virion once inside the cell, okay, is that virus gone? No. It's just still part of that replication cycle. So my favorite virus definition is unfortunately kind of a long one, but it has nothing to do with parasites. It has nothing to do with anything being very small. So here, viruses are entities whose genomes are elements of nucleic acid that replicate inside living cells using cellular synthetic machinery and causing synthesis of specialized elements, and so that's those specialized elements, or the virions here, that can transfer the viral genome to other cells. And so as far as I'm concerned, this is probably the best and most inclusive definition of viruses which are there. And it's also not a negative one. Viruses are not this. They're not that. I think it's very much better to have an inclusive kind of definition here. And this has withstood, as it were, the test of time. At least as far as viruses and virology are concerned. So this is getting back to your question, Ian, about what about the virion. So the process here, ceci est un virus, so this is the virus. It's this whole process. So a virion will infect the cell, release its genome inside the cell, that cell will now make more of the genomes, more of the virions, and this whole process then. So it's really the whole process. You can think of this big box around this and say this is the virus, not the individual virion. So that's the point about pushing the virion, that's only one part of this cycle. And if you're just thinking about the virion, that's not really your virus. And if you think about viral disease, a viral disease is not a virion. It's the interaction between that virus and whatever the host is, which is causing that particular disease. So this is really what that's about. So why do I study viruses? They're everywhere. We'll talk about this again in just a second here. But in seawater, which is actually the very best studied environment, there's anywhere between 1 million and 10 million virions per milliliter. And there are a hell of a lot of milliliters of water in the ocean. That gets you to an absolutely ridiculous number. Same thing is true of soil. Same thing is true of the air that we're breathing right now. Same thing is true of our blood, etc. So they're absolutely everywhere. They infect everything, even the crazy organisms that live in boiling acid, which is what we study in my lab, but pretty much everything else. I've always challenged people to try and come up with an organism that doesn't have a virus that infects it. There are even some intracellular parasites which also have viruses that infect them, which is really, I think, kind of amazing. Incredibly diverse, certainly in terms of structure, probably in terms of sequence as well. And the main reason, actually, that I got interested in studying viruses in the first place is they're great tools to actually understand the cells and the organisms that they're infecting. In fact, a very large number of virologists got into the field because they're interested in trying to understand some very fundamentals about how biology works by studying the virus. Because usually they're very simplified sets of genes and their regulatory processes, which can allow you to learn a lot about this. Yeah, and sure, they make people sick. And these are my favorite virus vectors. A little older now. This one's now 12 and this one's 9. But at least now they're trying to not be too much of a viral vector as far as moving things around. Yes, they get infected, but they also infect basically all of the other organisms that anybody's ever looked at in enough detail. For a long time, people said C. elegans doesn't get infected by viruses. 4 years ago, people found viruses infecting C. elegans. So it's basic. I think as long as you look hard enough, you'll find them. So hopefully everyone knows about this, the big tree here, looking at small subunits. Oh, yeah, sorry.
Oh, sorry. Could you predict that viruses were like the first to evolve?
Ah, so how old are viruses? I'm just going to paraphrase your question here. There's this bullet point right here. Some types are probably very old. How old? Did they predate cellular life? Well, if they're obligate intracellular parasites, they need a cell in order to replicate, so that whole process. So could they be precellular? Well, then you have to change your definition of a virus. So were there parasitic nucleic acid elements, or those that were piggybacking on everything else? Probably. Until we have a time machine, which I'm not working on in my lab, it's going to be very hard to answer that question. But we do have, and I'll talk about some of the evidence for some of these viruses being incredibly old, and probably at least as old as some of the, my pointer here is giving me trouble, the last universal cellular ancestor, which gave rise to all of the 3 domains of life as we currently know them. And I think there's pretty good evidence for that. Yeah, Angela.
Aren't there viruses that infect viruses?
Are there viruses that infect viruses? That's a great question. And if you think about the whole idea of thinking about a virus as that cycle, there are most definitely other viruses that will infect viruses in that process. Yes. Probably the best example is called the virophage that we'll probably talk about in 8 weeks. But yeah, there are some really fascinating examples of that. So yeah, everything gets infected by viruses. Between 10 and 40% of the human genome is viral. We can argue about some of the transposable elements, whether those came from viruses or the viruses arose from those transposable elements. And clearly, viruses are really important for evolution. I had a meeting again a couple of years ago now with a bunch of virologists who basically said if there weren't viruses on the planet, it would probably be covered in about a kilometer worth of slime, and multicellular organisms probably never would have arisen in the first place. So clearly absolutely critical for evolution. Viruses are everywhere. If you do take one of those samples of seawater that I talked about and stain it with a fluorescent dye that only will fluoresce once it binds to some kind of nucleic acid, this is what you see. One of the main reasons that I like this one actually has one poor, lonely eukaryote in it. This is a diatom down here. These big dots are bacteria and archaea. What are little dots? Yeah, or virions, probably a more specific way of talking about it. But yeah, absolutely ridiculously large numbers of these things. Again, people can do back-of-the-envelope calculations, anywhere from 1 million to 10 million per milliliter just in the ocean surface waters. That's 90+% of marine particles. So there are more viruses in a cup of seawater, just a cup, than there are humans on the planet. Just a cup, or virions, I should say. You do the back-of-the-envelope calculation, you get 10 to the 31. 10 to the 31 is ridiculous. That's just the number that I just can't comprehend. And it may be off by a couple of orders of magnitude. So it's only 10 to the 29. It's still a ridiculous number of these particles. And one way that you can, again, take this ad infinitum, an extreme, if you lined up a standard virus particle, say a bacteriophage like this one, end to end, 10 to the 31 of these would be 100 million light years long. So that's way out of our galaxy, well beyond the next galaxy. If you just line, all this is just the ones that are on our planet. Again, this is, to me, is just a little crazy. But how can you have so many? It may be a little hard to see here. That is actually my thumb up the top here. This is a 15 milliliter conical tube. And those of you who don't have the notes, how many virions do you think are in here? 10 to the 7th times 15. Any other guesses? A lot. A lot. So actually figure it out. There are 10 to the 11th virus particles in there. That's, what is that? That's 100 billion. This is just ridiculous. But it just goes to show, you can't even see them in here. And if I turn the lights out, this is completely clear. But there are 10 to the 11th infectious particles in there. Yeah. So is there a question here? Is there a hand up? Pardon? It is, but that's per mil. How many mils are there in it? About 10, actually, in this particular case. So, yes. So it is labeled 10 to the 10, but it's PFU per mil. Good that people are watching. I like this. So human genome, again, a lot of you have seen this before. The boring part, actually, no, the important part, sorry. I was going to say the boring part's actually the part over here, the unique protein coding sequences, the 1.5% of the genome. This gray part here, this is the part that nobody would argue are virus-like sequences. About 8% of the genome are clearly retroviral-like sequences, which are present in the genome. I would argue that all of the retrotransposons are related to viruses that are present. And this is just human genome sequence. Each and every single one of our cells has 40+% of the sequence which is related in some way to viruses. 8% are clearly obviously viruses, and up to 40% are like this. So you might wonder, what on earth are all of these virus sequences doing? Are they just junk, or are they actually useful in some ways? I'll give you one guess. Of course they're useful. They have to be useful. Otherwise, why would we hang on to some of them, evolutionarily speaking? Oh yeah, and that's that worthless piece down there as well. So it turns out that some of these sequences, particularly these what are called endogenous retroviruses, have contributed a number of genes that are really critical for life as we know it. My favorite example there is what's called the syncytin gene. Anyone heard about syncytin? Some of you may or may not have. Syncytin turns out to be absolutely required for the development of the placenta. If you don't have syncytin, you don't have a placenta. If you don't have a placenta, then we wouldn't have a syncytin, S-Y-N-C-I-T-I-N. If we didn't have a placenta, there wouldn't be live birth, so none of us would exist. There wouldn't be any motherhood. So viruses are important for motherhood. If we didn't have viruses, we didn't have motherhood. Whoa.
Or maybe we would have laid eggs.
Or something like that, which we may or may not have taken care of. And then there's also some marsupial aspects of things as well. So yeah.
You're saying endogenous genes, viral genes. So are those the non-retroviral?
Okay, so the question here, endogenous versus exogenous. So endogenous viruses versus exogenous viruses. So exogenous viruses would be a new infection which is happening, coming from the outside. The endogenous is an infection which has happened in the dim, dark, distant past. If we think about the syncytin gene, that's hundreds of millions of years that the first placental mammals came along. So there must have been a virus infection, an exogenous virus that infected that precursor of modern placental mammals that then was incorporated in the genome and kept in the genome and passed along since then. And so that's the endogenous retrovirus, is the one which is already in the genome and is being passed along. So those are then the ones that are being inherited on a vertical process. So an endogenous virus is present in the genome. Okay, I keep clicking through these far too rapidly here. So this is one of those cases where things have changed a lot, literally about the last 5 to 10 years. I always used to say the most genetic diversity that anyone's ever seen is present in viral genomes, because you've got 10 to the 31 virions that are out there. Each of them has its own genome. That's going to be a ridiculous amount of diversity, and that's certainly what it looked like at the time. If you sequenced viruses that infected mycobacteria, 50% of the genes that you found in the genome, this is a typical genome here, actually it's SSV genomes. If you just look at those marine samples, 90+% of those sequences didn't match anything that anybody had ever seen before. And if you went to these extremophile viruses, again, like the ones that we have here, 95% of the genes in here didn't match anything that anybody had ever seen. So you can do, again, some of these back-of-the-envelope calculations, and people thought there were billions of unique genes that were present in viruses. Now, it turns out that there are a whole bunch of unique genes, and we have no idea what they do. But when you do a very careful, much broader sampling, what happens is there are a lot of the same genes. So we don't know what they're doing, but there are a lot of them that are really, really similar to each other. And so some recent studies, and this is literally published about a month ago, have shown that actually it's probably not billions of unique genes, it's only millions of unique genes, but we really don't know what the vast majority of these genes are doing. So, great place to study, go and look at, try and figure out what a bunch of these unique genes are doing. How many of you are taking the mutant viruses from Hell Lab? So you'll be finding out about this and hopefully teaching us about it. So what are these unique genes doing? But even so, millions of unique genes are pretty amazing. And one of my colleagues, Roger Hendricks, called this the dark matter of sequence space. And you'll see why that is in just a second there. But it turns out that these much higher throughput sampling and this is the job that I want to have, sailing around the world collecting samples for viruses. They've been doing this for about the last 5 years now. And in looking at that, they only see about 15,000 different species of virus, and these are pretty common ones. And you would expect with 10 to the 31 virions that are out there, you'd expect it to be way more. But it actually seems to be a relatively low number. And we don't understand why that is, but the data are pretty good, at least for surface waters, which are the easy ones to get to when you're in your sailboat sailing around the world, which is what they did. Then you get only about 15,000 species. So these numbers here, unique genes, how much of phage genomes have actually been sampled, is really pretty up in the air right now. Lots of new genes. What they're doing we don't know, but not quite as ridiculously diverse as we thought they were in the past. So dark matter of sequence space, why do we call it the dark matter of sequence space? These are actually some data from my lab. If you look at sequences from a place called Boiling Springs Lake in Lassen Volcanic National Park, this is an amazing ecosystem. The low temperature is 50 degrees Celsius, about 120 degrees Fahrenheit. The high temperature is 95 degrees. It's a pH of 2, so it really is kind of a boiling acid ecosystem. If you look at virus-sized particles, I should say virion-sized particles in there, look at their genomes, about 7% of them match something, whereas the unknown sequences are over 90%. And this is actually kind of similar to dark matter versus visible matter in the universe. So this is our comparison here in the dark matter of sequence space, 7% versus 4%. So there's a lot of stuff out there that we know is there, but we really don't know what it's doing. So strange sequences, a whole bunch of little spots. What do those little spots look like? This is another case where you've got a huge amount of diversity. Virus capsids don't look like anything else. So where did they come from? Your question about how old are viruses? Well, if their capsids look really different, well, where on earth could they have come from? But interestingly enough, and we'll talk a lot more about this when we talk about virus assembly, most of them have this sort of icosahedral shape, kind of like a soccer ball. A few of them have these rod shapes, and a lot of them actually have a combination of icosahedral symmetry and sort of rod-shaped or linear symmetry. There are definitely exceptions. The pox viruses, this is an example of the myxoma virus virion. So these virions are geometrically constrained, and you don't see anything biologically that looks like these at all. But the most surprising is when people actually now took the electron microscope and cranked it up and started to look a few more of those little green spots. And what they found was in these extreme environments that you would see some things that kind of had an icosahedral structure with a rod-shaped structure. Some that looked more rod-shaped but had these really interesting claw-like structures at the end. Turns out this claw-like structure is really important for binding to the cell. This one, which was the very first purely icosahedral virion to be found in these extreme environments, and again, this is one that I found. We mostly work with these virions, the ones, again, that are rather like this, that don't look like an icosahedra or a helical form. Turns out these might actually have a structure similar to the inside of HIV, which is a really fascinating question that we're trying to figure out here. And probably my favorite virion is this one right here. This is the aptly named Acidionus bottle-shaped virus, because these really do have a bottle shape with what almost looks like a ring of candles at one end. These are tiny. These are on the order of about 100 nanometers in terms of their size. How the heck you can fit something like this together, or even for that matter, something like this together, is a really interesting question, and that's kind of what keeps me awake at night, trying to figure out what on earth is going on with these archaeal viruses. So that's what gets me excited about it. A number of other people get excited about viruses for other reasons. This is Salvador Luria's, and basically, why study viruses? study viruses if you want to understand the very fundamentals of life itself. So the simplicity of nature, what do we want to try and find? Contribution of science. Everyone marching on the 22nd, I hope? Yes, 22nd of April? Good. I'll be talking more about it later, don't worry. Unifying and simplifying generalizations, and that's what we're going to be really talking about for the rest of this term, is those unifying and simplifying generalizations. There's always an exception. This is biology. There's always going to be an exception or multiple exceptions. But what's most important, what we're going to be talking about, are these overall patterns rather than the individual kinds of aspects of them. Now, finish up with a couple of what I think are really open questions, and in fact, one of the ones that you brought up very well, thank you. Where do viruses come from? Are they precellular? Are they postcellular? How old are they? And why are there even still viruses around? If they're all parasites, you would think that 4 billion years of evolution would have gotten rid of them if they're really that negative. So one of the things that a number of people have looked at, virulent, meaning they're really nasty viruses, if you're killing off your host and you're dependent upon infecting a host, that's a really crummy strategy for keeping more of yourself around. So most of the really nasty viruses we'll see are either probably in a host that they shouldn't be in or normally aren't in. Ebola is a really nice example of that because, we end up dying when we get infected with it. Well, if you're dead, then how does that virus get to a new host? So these ones that are really killing things off really rapidly are probably new in that particular host or are just new in general. And probably there are some new viruses arising, and again, that gets back to the chimeric virus that we're talking about. On the other hand, these viruses, like the endogenous ones hanging out in the genomes, hundreds of millions of years. So, these things have probably clearly been around for a really long time and are probably actually not causing anything negative, and in some cases, maybe being positively selected for. So I think syncytian and the whole placental mammal thing is probably one as well. And then, are viruses actually good? I think that some of them are. You can check out this website that talks a little bit more about it, or you can also look at the trailer to my film, which I won't show because it's longer than we have for the rest of the time here, but feel free to go ahead and take a look at it. Somebody's making some money off of it because I actually started to get ads on it after 2,000 views. It's not me who's making any money off of it. But the kicker, as far as that trailer is concerned, just ignore the last, I think it's 3.5 seconds, which says coming soon, 2012. See you all on Wednesday.